Hi friends, welcome to Biogyan. Proteins are made up of L-amino acids except for a chiral glycine. This homochirality is necessary for the protein's proper folding and its functioning. Proteins cannot be a mixture of D and L amino acids. So in today's video we will be discussing why proteins are made up of only L amino acids. Why did this selection of L-amino acids for protein synthesis occur? While this question still remains difficult to answer, usually scientists agree that selection of L-amino acids for protein synthesis has been a chance event in the evolution. However, in the recent years, many explanations, theories, and experimental evidences have been put forward to explain this mere chance event of L-amino acid selection. We will be discussing here a few of the major theories of the selection of L-amino acids. One such theory is the existence of energy difference between the L and D peptides. The energy difference between the two enantiomers has been explained as parity violating energy difference. Parity in itself means similarity. This difference exists because of weak interactions. Weak interactions exist at inter and intra-amino acid levels. Calculations of parity violating energy difference reveal very low energy differences between the chiral isomers. However, the energy difference gets amplified as the amino acids start forming peptides. This energy difference leads to selection of lower energy form, which are the L peptides. From PVED calculations, it has been estimated that the selection of L peptides occupied a minimum time of approximately 1000 years. According to the theory of adsorption, D amino acid isomers were adsorbed onto the surface of materials like calcite, which were abundant in the prebiotic era. This left only L-amino acid isomers for polymerization, and hence the resultant L polypeptides got selected. This has been experimentally proven by many, where racemic mixture of a certain amino acid were exposed to calcite crystals, and the result was adsorption of D isomers on the calcite crystals, leaving behind only the L amino acid isomers in the solution. Unfortunately, the adsorption of all amino acids racemic mixture was not the same. A similar theory of preferential crystallization of D-isomers has been proposed and experimentally proven. According to this theory, in the prebiotic soup, D-amino acid isomers were abundant. As the soup started cooling, the excess D-isomers began to crystallize and precipitate, and then only L-isomers were left behind in abundance in the cooled soup. Our galaxy has a characteristic chiral spin and magnetic orientation, which results in cosmic dust particles to polarize sunlight as circularly polarized of a particular orientation. In the prebiotic era, this circularly polarized light may have degraded D isomers preferentially, resulting in the abundance of L isomers and hence resulting in their selection. Ribonucleic acids have been proposed to play an important role in the chiral selection of proteins. Ribonucleic acids were proposed to be present before the synthesis of first proteins started. RNAs were chiral selected to use only D-ribose in their sugar backbone. The first process between an amino acid and RNA is amino acylation of the RNA. Thus, RNA must have shown preference for L amino acids. Of note here is that RNA molecules did not exhibit stereoselectivity in free solution when this was done experimentally. Only when the RNA was constrained on a surface which resembled surface monolayers in the prebiotic era, it started exhibiting preference for L amino acids. Stereoselection by RNA has been proved experimentally by creating a RNA mini-helix, which is believed to be progenitor of the modern-day transfer RNAs. RNA mini-helix synthesized using D-ribose sugars showed preference for L-amino acids. However, if L-ribose sugars was used to synthesize RNA, then the opposite was true. L-ribose containing RNA showed preference for D-amino acids. Sugars like amino acids also exhibit chiral bias. Most of the sugars are made up of D-isomers. Thus, RNA contains D-ribose, which exhibits preference for L-amino acids. Thus, we can say that chiral selection of sugars played an important role on the selection of L-amino acids. Also, chiral selection of these sugars might have been completed before the selection of L-amino acids started, or the selection of these sugars might have been initiated prior to L-amino acid selection, and the selection of both amino acids and sugars might have overlapped for a certain period of time. A certain chemical reaction is known to exhibit autocatalysis when the product of the reaction is a catalyst of the reaction. The autocatalysis results in the amplification of a small difference in the abundance of stereoisomers. Choi in 1995 has demonstrated this effect and has found a small difference in the abundance of stereoisomers, which can be amplified to yield abundant amounts of a particular stereoisomer. These reactions could have been possible after simple organic molecules started appearing in the prebiotic era. To summarize, why only L peptides got selected cannot be answered by only one of these theories. It may be assumed that a number of selective pressures might have acted at the same time or in a stepwise manner or in a combination thereof, selecting the L peptides. Also of interest to note is that meteorites have been found to be rich in certain L-amino acids, thus giving boost to the theories of extraterrestrial origin of life. Here are references for your further readings. Please like, share and subscribe Biogyan. Thank you.